Hello, my name is Erich Jarvis. I'm glad to be part of this symposium and presenting work that my lab has been doing on COVID-19 research in relation to genomics. And just to give everybody a little bit of background, I'm actually a neurobiologist and a genome biologist, and I am running the Vertebrate Genomes Project. I'm chair of it, whose mission is to generate high-quality genome assemblies eventually of all vertebrate species. Right now we're doing all vertebrate orders. And with the ability to produce high quality genome assemblies, I got involved in this structural variation consortium with the idea to find out whether or not we could find greater structural variations in people who have severe COVID reactions compared to those that do not. And to use that technology to address this question in collaboration with Bionano and others. And today I'm going to tell you about a story on the Interferon-1 alpha and beta receptor 1, that there's some variation in that receptor, we think, some preliminary findings that correlates with degree of COVID-19 severity. So the data set that we're working with actually comes from Bionano, the initial data set, a metadata approach. Here we have hospitalized patients, those who have been severely ill in ICU or have died, 30 of them, and controls, a random set of controls that are in the Bionano collection, 57 individuals. Bionano generated chromosomal length Bionano maps of these persons, aligned it to the human GRC H37 reference genome, and called variants for each haplotype, not just one haplotype, but for both haplotypes throughout the genome. And that's important to focus on both haplotypes. And initially, just looking at these variants in the hospitalized patients, we found some interesting genes that are highly represented with more variation than you would expect by chance in some immune genes and respiratory system genes, and actually even genes that are involved in coronavirus interferon induction. At a significantly high level of enrichment of gene ontology categories. But when we looked at the controls, we actually found some of the same gene families that were showing greater variation than usual, than you would expect by chance, and also in the controls. And that then led us to realize, well, these genes, including immune-related genes, are genes that actually do have a lot of structural variation in them to begin with. So we can't use this as a criterion as, compared to the rest of the genome background to ask if there's a difference between the severely ill patients and those who in the general population background. But we did find something else interesting when we started to look at the actual heterozygosity. And that is for this interferon alpha and beta receptor 1 subunit, we found that there's some variation in the upstream promoter region of this gene in terms of insert size. And we found that the control group, roughly 20% of them have large heterozygous variation in this region. Whereas in the hospitalized group, an amazing 78% or so of these 37 patients had high levels of insert size heterozygosity in this region. Whereas in contrast, another gene involved in the immune pathways, interleukin-1 receptor type 1, where both roughly 60% of the control and the hospitalized group have heterozygous variation at a similar level as what we're seeing over here for this interferon alpha beta receptor. So, so what this meant is that there's something different, not about the actual individual the individual variant, but the level of heterozygosity of that variant. So we actually then aligned the Bionano maps back to the assembly and try to figure out what exactly is that variant that we're seeing. And what we're seeing is that there is this highlighted in this red circle here, in front of the 5' region of the gene, there is a repetitive a tandem repeat structure of nucleotides that are varying from one person to another and also one haplotype to another. And what we see with the patients who become severely ill is that the variation of those two different haplotypes in this tandem repeat is greater. And we can map that out to show here that there's variation in the length of this tandem repeat when you add up both haplotypes together. But and you can see a difference here between the control and the severely ill patients. And so what we're going to do, and so putting all this together, going back to this plot here, is what we think is going on is that the greater heterozygosity might be causing a deficit in the function, let's say, of the regulation or the function of the first intron of this receptor, that in its more heterozygous state, where the two different variants are more similar to each other, is not as detrimental to, let's say, the function of this gene. And so this is consistent with this interferon gene pathway being found involved, or at least other genes like the interferon molecule itself from the Casanova group and the consortium that he's helping to lead, that different genes in this same pathway are being found to be associated with the degree of severity of this disease. And we think this is one more example. And to show you in a cartoon fashion, this diagram from Vertebrate et al. Shows that when the virus binds to a cell, it binds to the ACE2 receptor, it gets taken up, that causes an immune response in that cell to release the interferons. They bind to these receptors that we're finding this difference in heterozygosity, and recently also the interferon alpha-2 receptor, a publication in Nature a few weeks ago showed that there's some variation there that's associated with degree of severity. And these receptors can dimerize here. And we think that the heterozygosity is affecting that level of dimerization to cause a differential immune response in cells that respond to the infection. So future efforts, what we're going to do is we're going to generate more complete de novo chromosomal level reference genome assemblies of COVID-19 patients, 20 persons who have been severely ill, 20 controls who are asymptomatic individuals matched for age, sex, and pre-existing conditions. And we're going to use the vertebrate genomes pipeline for this, where we have PacBio long reads, bionano optical maps, and high-C data from various groups like Aromagenomics and Dovetail. And we're going to use these data types to produce as complete a genome assembly as possible, and then interrogate this region of the genome to find exactly what kind of variation we're seeing in heterozygosity and other types of variants. And we're going to compare the human data with data from 260 vertebrate species that we're generating high quality data for, representing all vertebrate orders, including species that are more susceptible and less susceptible to being infected by COVID-19, to see if there's any correlation with humans who show greater susceptibility to being sick from this virus. We're going to do that from the nucleotide to the structural level of analysis on human and non-human animal genomes, and identify the exact kind of structural variation we're seeing in these cases that I just showed you in the previous slide with the IFNAR1 gene. And if you want to join this effort, I encourage you to contact me. And before I finish up, I'm going to go right back to the beginning to thank my colleagues here, Farouk, who is a graduate student in the lab, who did a lot of the analysis, Olivier Fedrigo, who helped also with the analysis, who's a bionano expert in our lab, Alex Hasty, who works with bionano, who also helped us out with these analyses, and myself, who's running or supervising this project. And I will go ahead and stop there, and thank you for your attention.